Here we go. All right, welcome y'all to the first weekly review session. There will always be some sort of handout available. To access it, you'll click on the Lecture Unit 1 module, go into the unit that we are discussing, go all the way down to the Resources folder, and there will be a Weekly Review Sessions folder that I just added, and inside of that is going to be your handout. So go ahead and make sure you download that and have it handy. While people are doing that, and while I get it up on my screen, I do want to mention two things. From here on out, your knowledge checks are always going to be on Thursdays and Mondays, okay? Thursdays and Mondays. They're not going to kind of be on random days. So I know this past week, the checklist said Wednesday. I had forgot to actually program it into Blackboard. And so some people didn't even see it because it wasn't showing up on like y'all's calendar and stuff. So if you did it Thursday, that was fine. But from here on out, knowledge checks will always be Thursdays and Mondays. At this point, you should be able to see the knowledge check questions. So when you go into your grade book, it'll show you the score you got. You click on that score, and it's able to take you back into the quiz and show you the questions. And it should tell you whether you got it right or wrong. But the answers are not visible. And that's because in the past, things end up on Quizlet immediately. So then future students don't actually have to study or know the material to get perfect scores on things. So it's just something that a lot of us in the department do. We want you to see the questions so you can go look up the answers or come to me and talk about the questions, but they're not just there for it to kind of be copy and pasted online. So if you ever have questions about knowledge checks or quizzes or anything, please don't hesitate to let me know. Okay, here is the week 1 review session. Let me see, where are y'all? I have too many. I have 3 monitors set up and they're all going a little wonky. Can y'all see that? You cannot see that. I swear, this is not my first time using Blackboard. It's the share screen option. We used to have one option when you pushed that. Which screen do you want to share? Now it's which tab, window, this, that. It's like, for Pete's sake, just share my screen. All right, here we go, here we go. Okay, so we are going to talk about the intro to micro and cells, which I know is a lot of information, but y'all, next week is going to be insane, okay, because you're going to be talking about genetics. So DNA replication, transcription, translation, viruses, all these big things. That's not a lot of time, right, for a bunch of new content and old content. So how I usually run these reviews is we're going to do something like you see on the screen, number 1 through 15, which is meant to just kind of get these words, these people, these ideas back into your head. Then we're going to move on to stuff where I ask you to tell me things. And I want you to try and answer this stuff without your notes at first. Because I'm going to say this to y'all all the time over 8 weeks. Using your notes as a crutch is the biggest issue that students report to me. Because it's not surprising, right? Having that kind of safety net. You want to use your notes. They're right there. And while it's good to use your notes because it validates, like, oh, hey, the answer to this question, she did tell it to me at some point, right? That's great. You might get a perfect score on that knowledge check or unit quiz, and that's great. But on those high stakes assessment, the tests, you don't get any notes. You don't get a cheat sheet or a blank sheet of paper to do a brain dump. All you get is your brain. So I am going to constantly be encouraging you, okay, try to answer these without your notes. It's okay if you get it wrong. This is the place to get it wrong. And then I'll have you go back and use your notes, modify your answers, things like that. So when I say don't use your notes, please try not to get annoyed with me. It is for good reason. I want you to see what's stuck in your brain already and what's not. That way you don't waste time studying things you already know, but you also don't move on to DNA replication when you don't have the scientists nailed down quite yet. okay, because this unit will snowball. Before you move on to new stuff, you're going to want to have this stuff committed to memory, okay? All right, let's go ahead and start. We're going to do the first 5, and here's how it's going to work. I'm going to read the statement, and then if you guys will go and open up the chat, that little chat bubble down on in the little menu bar to the bottom left, and then put your cursor in the box that says say something. And I want you to put T if you think the statement is true, F if you think it's false. But don't push enter until I say go. All right. So I'll say, okay, number 1, blah, 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 blah. All right, y'all. 1, 2, 3, go. And on go, you push your answer. Here's why. It causes you to just see what everybody has said without y'all paying attention to who said what. Okay. And that way your answer is not swayed by what everybody else has said. Because again, this is the time to be wrong. But if you're right, hey, you know it, good job. Next, okay? So again, I'm gonna read the statement, I'm gonna wait a few seconds, and then I'll say, okay, 1, 2, 3, and on go, you push enter. All right, here we go. True or false? A bacteria will always have a chromosome and sometimes a plasmid. Type your answer in, but don't push enter. All right, T or F, 1, 2, 3, go. There we go. Excellent. Okay. So this one is true. Now, whenever you see the word always in science, you need to make sure you read it a couple of times. Very few things are always. But chromosome is a fancy word for DNA. All living things have DNA, right? That's the genetic material. That's what makes us who we are. Can somebody tell me what a plasmid is? You can either come off mute or you can type it, whatever you prefer. Don't be shy. This is something that a lot of people tend to forget. So in case you're typing it, keep typing. I will go ahead and answer it, but brace yourselves, y'all. I'm going to ask y'all for a lot of information in these. So remember, a plasmid is basically a bonus feature. It's an extra piece of DNA. Yep, it does come back in the genetics section, absolutely. So plasmids are extra pieces of DNA. It is separate from the chromosome, okay? So all the information on a chromosome is essential. You must have it to be who you are. Plasmids are bonus features. They are not essential. If a bacteria has a plasmid and loses it somehow, no problem, it's okay. So I'm going to put some words in the chat, and you're going to want to associate these with plasmids. Let me see if I can type without too many typos. Chromosomal. Extra chromosomal. So notice chromosome is there. That means the DNA that's required. Extra means it's separate from. So plasmids are not part of the required DNA. They are bonus features, okay? So they are non-essential. So when we get to like antibiotic resistance, some bacteria are resistant to certain antibiotics because of a plasmid. That plasmid may code for an enzyme that breaks down penicillin. So they can survive without that, but it's definitely an advantage, right? Okay, so I get very excited about these topics, so I'm gonna try to rein it in since we got a little bit of a late start, and I apologize for that. Normally we start right at 10. All right, Virchow developed the theory of spontaneous generation. Put T or F in the chat, but don't push enter. If you want to, anytime the answer is false, feel free to put the word that would make it true if you want. All right, true or false? Virchow developed the theory of spontaneous generation. All right, 1, 2, 3, go. Excellent, good job. So Virchow developed the theory of biogenesis, which states that living things come from other living things. And so he didn't offer any extra evidence to support this, but he was trying to give people something to hold on to, right? We've learned a lot the past 2 years. People don't just abandon their beliefs because you tell them to, right? And so even though Reddy and Spallanzani had given evidence to suggest that spontaneous generation, basically the poof, here something is, right? Magic. To prove that that's not really a thing, people didn't have any other theory to accept. So we were asking them to abandon spontaneous generation, but not really offering any alternative, okay? So that was Virchow's big contribution, is because he said, hey, look what Reddy and Spallanzani and others have done. Maybe this can help explain it. So it was pretty big deal. Good old Virchow. All right, number 3, a fungal cell may be described as bacillus. True or false? 1, 2, 3, go. All right, this one is going to be false. A bacterial cell is going to be described as either bacillus, coccus, or a spirochete or spirillum. They're two different types of spiral-shaped cells. And so remember, we focus on bacteria in this class, and in lab, we talk a lot about morphology and arrangement. And I have a question about that in just a few minutes. But whenever you hear bacillus or bacilli, that's plural, coccus or cocci, and then spirillum or spirochete, there's not really a plural form of that. That's always for bacterial cells. All right, number 4. This one's a bit harder, so take a second to think about it. A eukaryote uses a pilus to grab hold of another cell. True or false? Don't push enter yet. Take a second to think about it. Don't lick your tongue. All right, on 3. 1, 2, 3, go. Starting to see a pattern, right? I like to give y'all false ones. Yes, this one is also false. Pilus does in fact grab hold of other cells, but a pilus is used by a prokaryote. This is a prokaryotic structure. So that's going to be one of the hardest parts of the cell's content is, yes, you need to know what each structure does, but you also need to know if it's associated just with prokaryotes, just with eukaryotes, or both, okay? So for instance, prokaryotes and eukaryotes are all living things, right? So especially when you're doing your week 1 activity, that overlap section, saying what do they both have, basically what you're asking yourself is, do all living things have this? So we've talked about chromosomes, DNA. Every living thing has DNA. It's a requirement, right? And so that's something that you would put in that middle section of your Venn diagram. But a pilus is just found on prokaryotes. And in the genetics section, you'll learn more about what it does, but it's basically going to grab hold of a neighboring cell and it's going to share its DNA with it. It's like a form of, like, it's not non-sexual reproduction, but kind of. It's one of the ways bacteria can share their DNA with other bacteria. It's pretty snazzy. All right, number 5. Bioremediation is one way microbes harm people. So in other words, bioremediation is bad. T or F? 1, 2, 3, go. I'm seeing a trend here. I promise there are true statements in here somewhere. Yes. So bioremediation is good. It is helpful. So think of like oil spills, right? So remediation means to fix. Bio normally means like biology, like living things. So bioremediation is using a living thing to remedy a problem. So with oil spills, there are bacteria that legitimately eat oil for dinner. And so by introducing those bacteria to an oil spill, it can clean up that oil without disrupting the ecosystem. So bioremediation is a very beneficial thing. All right. Number 6. This is something that is going to be really important for your unknown report as well. So we're going to spend a little bit of time on number 6. You observe a cluster of circular cells. This morphology and arrangement is streptococci. True or false? Streptococci. All right, on 3. 1, 2, 3, go. I know y'all are like, this lady never gives us true. I do, I promise. Good. Now, can somebody tell me what is the correct morphology and arrangement for a cluster of circular cells? Let me see if this will let me draw. So remember, y'all, a cluster of circles, I'm drawing with my mouse, so it's extra bad, sorry, would be kind of like this. Hey, good, Jessica. Yes, staphylococci. Yep, staphylococci. Excellent. So y'all, here's kind of a thing that I see a lot on the unknown report. So you will hear this later. Morphology is written, morphology, let me erase that. Even though I always ask, give me the morphology and arrangement, notice that it's written as one word most of the time. And morphology is listed first, but it's actually the second half of that kind of merged word. And so morphology is always going to be the cocci, the bacilli, things like that. The arrangement, ooh, that was thick, sorry. That's going to be the first part, okay? So a cluster, that's the arrangement. And so that would be the staphylo, okay? And then circular cells here, trying to color code, because this is a very common point of confusion. Circular is the morphology. The science term is cocci, okay? So anytime I ask for morphology and arrangement, you always, always, always give me the science term. I cannot tell you how often on an unknown report, somebody will tell me it's a cluster of circles. You don't get any points for that, y'all, because that's not micro terminology. I say it sometimes in reviews and on the videos to try to help you understand that a cluster is staphylo. So I'm trying to kind of familiarize y'all with the common term and the science term. But on assessments, you always use the science term. So what would streptococci look like? So the cocci would still be circles. Exactly, Paula. It'd be a chain of circles. Okay, so the circles, again, and I'm sorry, I'm not color coding anymore. The circles are cocci, and then the chain is going to be the strepto. Now, coccis, remember, they have a lot more diversity in terms of arrangement. We can have a single cocci, we can have diplococci, and then we can have the strepto and staphylo that we've already talked about, and we can have tetrad. And there are actually more, but there's no need for us to go into all of them in this course. So a tetrad is just 4. And the reason these cocci cells can have so many different arrangements is because they're basically a perfect circle. And circles are symmetrical on multiple dimensions, right? And so let me go back to a thinner color. This little cocci cell here, it can split in half this way. So if you kind of look at me real quick, like that little camera. So if this is a cocci cell, pretend that's a circle, it can split that way, or it can split this way, because it's exactly the same, splitting right to left or top to bottom, right? Because it's a perfect circle. So lots of options. Bacilli, y'all, not as many options. Which arrangement can a bacillus cell not make? Does anybody remember? Tetrad is one, good. So there's one more, but let me explain why. Pretend y'all that I know how to draw. So if this is a bacillus cell, it can only stretch and pinch off. So it can only divide end to end, okay? It cannot divide up and down like that. It's just not possible. Okay? It can only stretch and separate. So that's one reason tetrad is not an option. Which other arrangement involves cells kind of growing on top of each other? Staphylo, exactly. Staphylobacillus is not a thing. Impossible, okay? So just something to keep in mind. So if you had just this, let me try to erase the others. If this is what you saw under the microscope, just a bunch of bacillus cells hanging out by themselves, in that case, your morphology and arrangement is single bacilli, and that's two separate words, okay? So if single is the arrangement, that's the only time you will write things in two different words. Single cocci, single bacilli. Any other time, you merge them, okay? So let me emphasize that. This is like the biggest mistake I see on unknown one, and I mean overwhelmingly. So that's kind of why I'm beating it to the ground here. So if instead you saw this, what's my morphology and arrangement? Don't be shy, y'all. We've got two bacillus cells stuck together. Excellent, Paula. Diplobacillus. And I'm not a grammar person, so if you say diplobacillus, diplobacilli, both are perfectly acceptable. Honestly, it's whatever comes out of my mouth is kind of what I say. So notice what Paula did here, y'all. She merged the arrangement and the morphology. It's one word. That's exactly how it should be. The arrangement is the first part of the word. The morphology is the second, okay? There are fill in the blanks, Jessica, but I don't ask you to do like crazy words. And I do go in and look to see, like, if you're close, I'm going to give it to you. So don't waste your brain energy, like memorizing exactly how to spell all these things. Good question. Yes, Melissa. So like, let's say it was a single bacillus. Here's how it would go when you write it. Two separate words, but you have to say single. Don't just put bacillus. Because if you just write bacillus, I go, oh, cool. Melissa knows that they're rod-shaped cells, but how are they arranged? Does that make sense? Are y'all feeling okay with morphology and arrangement? Can y'all give me like a yes or a thumbs up or something, just so I can kind of gauge? And if the answer is no, tell me that. All right, all right. And just FYI, y'all, I have to warn my in-person class of this too. There's a lot of times I'm going to say, does that make sense? And somebody geniusly made a meme of that. And I don't say that. I don't say, does that make sense? Because I'm questioning your intelligence. I ramble sometimes, and I'm afraid that sometimes I get too wordy and my goal gets lost. So it's just to give you an opportunity to be like, whoa, you lost me. Try that again, okay? I will never be offended if you're like, nope, I'm gonna need you to be more concise. That's just me. Thank you, Paula. Okay, so I know we're at 10:30, so I'm gonna go ahead and keep moving us along. And if we do need to stay a little longer, that's fine. It's recorded, so if you can't, of course, these are never obligatory, but me and my technical difficulties slowed us down a little bit. Okay, number 7. Reddy used decaying meat to try and disprove spontaneous generation. True or false? And at this point, just go ahead and type it in. Don't be shy. Nope, this one actually is true, y'all. Remember, Reddy put decaying meat in 2 jars, one that was open and one that was sealed. And his big finding was there were no maggots and flies on the sealed container. So if spontaneous generation were true, you'd see flies and maggots everywhere because they would have just poofed inside the jar. Okay, so Reddy's big argument was, look, there's only fly and maggots when it's open for them to get in. When it's sealed, why aren't they there? If they can just appear, why are they not there? Okay, yeah, there's so many to keep straight. Okay, so I would kind of make a note because there are so many scientists in this spontaneous generation debate. Reddy is against, he is anti-spontaneous generation, okay? And we get to some of the others here in a minute, so we can kind of review the rest if we need to. So Reddy he tried to disprove it, okay? All right, number 8, both bacteria and archaea are unicellular organisms. 1, 2, 3, go. Yes, there we go. Good, true. What else do bacteria and archaea have in common? Yes, Randy, good. They are both prokaryotes. So I know you guys are writing those statements, right, as part of your week 1 activity. And so a good piece of information to include is if it's a prokaryote or a eukaryote. And here's why. If you say it's a prokaryote, I know, and anybody else should know, that prokaryotes automatically narrow down the options to bacteria and archaea. If you say eukaryote, you've eliminated 4. You've eliminated bacteria and archaea, but you've also eliminated viruses and prions because viruses and prions are not considered to be alive. So we only consider living things as either prokaryotes or eukaryotes, okay? So by saying eukaryote or prokaryote, you're actually giving people really big hints, okay? And so that's just a suggestion. You don't have to put those in your statements. If you're doing a virus or a prion, part of your hint could be, I'm neither a prokaryote or a eukaryote, because that takes us down to 2, right? Virus and prion. So your other piece of information would have to help us figure out which is which. Does that make sense? I know those statements can be a little challenging to write, and you are welcome to send me a picture of your statements on Remind. I've had somebody do that. If you're like, am I on the right track here? Does this make sense? I'm happy to take a peek at them. Okay, now I'm not going to grade them all, but I am happy to look at them. Okay, let's keep moving. Number 9, a pathogen is an organism that does not cause disease. True or false? Ready, go. Sorry, I forgot about that part. Good, that's false. You're going to hear the word pathogen probably more than any other word in this class. A pathogen is a disease-causing entity. Notice the word I chose, y'all, because prions and viruses are pathogens, right? They make people sick, but they're not organisms. So I used the term, in this case, entity. Okay, now you can use the term loosely and say a pathogen is an organism that does cause disease, because bacteria, helminths, all of those, they are organisms. But if I were trying to be like as scientifically accurate as possible, I would say a pathogen is a disease-causing entity. Okay, because again, you kind of have to be careful with viruses and prions, and y'all will learn all about them next week. All right, number 10, the formation of biofilms is one way microbes benefit humans. Biofilms are good, is what this is saying. Ready, go. Good, excellent. Okay, so biofilms are bad, okay, and you learned about the capsule. The capsule is the structure that allows biofilms to form. Remember, capsules are sugary. Oh, I'm already answering a future question. That's okay. Capsules are sugary and sticky, so it lets some bacteria stick to a surface. They then recruit other organisms, bacteria and other things, to kind of hang out with them. They form this little community. I actually had my in-person people change their notes. Next to biofilm, I think I put mass of cells, which is technically true, but I prefer community of cells because it's not just bacteria. It's all other types of organisms that hang out together. And they're problematic because they clog things. Okay, they clog heart valves, they clog catheters. Okay, but they're also more resistant to antibiotics. Because if you remember, a biofilm is covered by this little protective hydrogel. They release this little gel molecule that surrounds them, so it's harder for antibiotics to get through and actually kill those bacteria. So biofilms are a huge headache for humans. All right, number 11, cilia are hair-like projections that cover some eukaryotic cells. True or false? Ready, go. Good, this is true. Cilia are specific to eukaryotes, okay, at least how we're talking about them. They are these little hair-like projections that help eukaryotes move around. They kind of, the cilia have this like swaying motion, and so it pushes the eukaryotic cells around. Now, bacteria have short hair-like projections, but those are called fimbriae. So fimbriae are only on prokaryotes, but their role is attachment or adhesion. Okay, so they look similar. Fimbriae and cilia look similar to each other, but cilia are on eukaryotes, fimbriae are on prokaryotes, and their job is very different. Fimbriae are for attachment or adhesion, same thing. Cilia are for movement. All right, number 12, a capsule is made up of sugar and helps pathogens evade our immune system. That means it helps it escape our immune system. 1, 2, 3, go. All right, only a couple people answered. This one's a bit trickier. So there's a lot to this one. So a capsule is made up of sugar. That is true. Okay, that's why it's sticky. Does it help pathogens get around our immune system? Yes, it does. Because you may have learned, for those of you that took A&P, the process of phagocytosis, which is where our cells go and engulf and destroy pathogens, right? It's how one way our immune system clears an infection. Well, our phagocytic cells have to catch the bacteria. The first part of that is grabbing hold of the bacteria, like a game of tag, like, oh, I got you. But because capsules are so sticky, sometimes the phagocytic cell has a hard time grabbing hold of the pathogen correctly. It's just like when a kid is covered in like candy and sticky stuff and you go to grab their hand and you're like, oh, what is that, right? Oh, that's what happens with our phagocytic cells as well. So the capsule is an advantage to the bacteria because it makes our immune cells have a harder time grabbing hold of them and getting rid of them. Okay, so capsules, we're going to talk so much about those in this class. All right, number 13, Needham attempted to prove spontaneous generation by boiling broth in uncorked flasks. Ready to go. I like the question mark. I'm with this one, and here's why. And this is why I read y'all's exams like 5 times, because this probably isn't my greatest question, and here's why. So when I wrote this, it was intended to be true, but I can see why people said false. So the first part is true. Needham supported spontaneous generation, okay? He's on the yes team, okay? So remember what Needham did, and I can't draw, y'all, so brace yourself. It was a round-bottomed flask, and he filled it with broth. My in-person class loves how I draw fire. Here we go. A flame. Use your imagination, okay? Can't draw at all. And so what Needham did is he put beef broth in this flask. He put it over a Bunsen burner so it boiled. He boiled it for a while. But then what he did is he let it cool, and then he corked it. And so, of course, things grew in that tube because he let the broth cool. When the broth was cool, all the microbes in the air fell into it. So when he corked it, those things were trapped in there. They ate the beef broth, they grew, and Needham was like, see, aha, I boiled things, I corked it, I win. Yay, spontaneous generation. But that's where Spallanzani came back. And I love that name. It just sounds sassy, right? I can just see this old dude being like, no, bro, you did it wrong, right? Because remember, Spallanzani came back and did almost the exact same thing. He took, let me see if it'll let me draw off to the side. He took, yes, oh, nope, okay. He took his own flask that was identical to Needham's. Sorry, I'm squished here. He corked it to begin with, right? So it was sealed from the get-go. Then he boiled it. And then once it cooled, nothing ever grew because there was no way for microbes to get back in. The broth was too hot. Does that make sense? So Spallanzani was against spontaneous generation, and his entire goal was to poke holes in Needham's experiment because it was flawed. Okay, so far, Reddy and Spallanzani are anti-spontaneous generation. Needham is pro-spontaneous generation. And who's the scientist that shut down spontaneous generation for good? Oh, y'all can just tell me. Does anybody remember who shut down spontaneous generation completely? It would be Pasteur. Pasteur did. Yes, good, y'all, good. Because remember, this setup, Needham and Spallanzani, we're on the right track, right? By boiling things, you're killing anything that currently exists, and then corking it, right? Whether it was before or after, like the goal was good, the intention was good. But remember, people don't want to let go of their beliefs. I mean, and it's kind of understandable, right? But what happened here with Spallanzani, so this one, remember, he put the beef broth in, he corked it, he boiled it, nothing grew. The biggest reason people didn't want to believe it or weren't convinced by it is because of oxygen or lack thereof. At that time, people assumed all microbes are like us. We need oxygen to live, right? So they all assumed, well, microbes must too. They're alive. They need oxygen. Okay, we now know that's not true. Plenty of microbes are actually harmed by oxygen. More on that in Unit 2. And so their biggest complaint about what Spallanzani did was, well, of course nothing grew. You corked that flask so no oxygen could get in. So Pasteur he designed essentially the perfect experiment. He also had a flask, but it was an S-necked flask, and that's worth remembering. I'm going to do my best to draw it. ACC should really hire me to teach art, right? Oh, see, I just messed it up. This part should not be closed. There we go. So Spallanzani had a very similar setup, right? He put all this beef broth into a flask of a similar shape, but because of this S-neck, it was open, so oxygen can get into the system. This actually lets microbes get in too, right? So let's pretend these are microbes that are in the air. They're going to fall in here, right? But microbes don't have little legs. They cannot climb all the way up this tube and dive into the broth. They can't do that, okay? So if microbes did get in, they were just stuck right here. They couldn't actually reach the broth. So when he then boiled it, he let it cool, and nothing ever grew. And he said, see, y'all, even when oxygen is allowed in, there's no growth because we've boiled it and microbes couldn't get in. Of course, there's always naysayers. So Pasteur had a flair for the dramatic. So what he then did is he said, okay, do y'all see how this cooled beef broth, it's been chilling here for a week now? There's nothing growing in it. He then broke the flask right there. Not quite there, but he broke it in such a way, kind of like this, but now it's open to the air. By the next morning, it was full of microbes. And he said, see, y'all, there are microbes in the air. When I allow them to get in, they will start to grow. So in this one experiment, he shut down spontaneous generation. He proved that that is not a thing, but he also proved that microbes are in the air, because as soon as he broke that flask, within 12 to 24 hours, there was growth. All right, are we feeling okay with Spallanzani, Needham, and Pasteur? Oh, it's an S-necked flask. I'll put that in the chat. S-necked flask, because the neck has an S shape. All right, are we all feeling okay with some of these scientists? I know there's a lot, and sometimes their experiments aren't terribly different from one another. All right, I'm going to take y'all's silence as being okay. All right, number 14. Protozoans are prokaryotes. True or false? Ready, go. Yeah, so quick. Look at y'all. Good. False. Remember, only bacteria and archaea are prokaryotes. Protozoans, algae, helminths, and fungi. Am I missing one? I might be missing one. But those are eukaryotes. And then again, viruses and prions don't get either classification because they're not alive. All right, y'all, you may have noticed from the videos, I love flagella. I just think they are the goofiest, most functional structures that help things move around. So you definitely want to know how we describe bacteria based on flagella. So a bacterium with flagella at each end of the cell is classified as amphitrichous. True or false? Ready, go. Good. Yes, that is true. Amphi means both. And trichous, I think, is Latin for hair because they look like these pieces of long hair. So amphi means both. So there's hair or flagella at both ends. Okay. Remember, the whole point of flagella is to move around. Okay. And you find flagella on both prokaryotes and eukaryotes. Their structure is a little bit different, but it's a form of motility for both types of organisms. All right, I've got like a whole page that we haven't even gotten to yet. So I'm going to go ahead and move a bit faster. So this is a really big idea right here, gram positive versus gram negative. So I'm going to give you just 30 seconds. Try to just use your brain. Write down what you find in a gram positive cell wall and what you find in a gram negative, because I promise, come exam day, you need this burned in your brain. Ready, go. Take about 10 more seconds. Just see what your brain remembers. Okay. What is the main component of the bacterial cell wall? Somebody type it into the chat for me. Not quite. It does have some, and it starts with a P, so you're on the right track. It's kind of a weird word, though. You are getting close. Melissa, but here's why I'm loving your answers. A, because you're giving me answers, and I can work with that. But B, peptidoglycan. The peptido part means peptide bonds. So there is some protein component there. Glycan means carb, and you gave me glyco there, okay? So you're right. Peptidoglycan is kind of part protein, part carbohydrate, okay? So excellent. You're totally on the right track. So when you hear cell wall, your brain needs to go peptidoglycan. And that's a weird word. Say it like just 10 times in a row, like right before you go to bed. If you haven't figured out yet, it took me a long time to learn this. Trying to memorize things right before bed, normally you'll wake up the next morning knowing that material. Now you got to do it in short pieces, but if you say peptidoglycan like 15 times right before you fall asleep, I bet you can say it perfectly in the morning. Worth trying. All right, so peptidoglycan is the big thing that we need to talk about. And I know we're running out of time, so I'm gonna be less chatty Cathy. All right, so gram positive, they have lots of peptidoglycan, thick layer of peptidoglycan, and that is good. That is positive, okay? Because peptidoglycan is just a really strong structure. It's going to maintain the shape and integrity of the cell. Peptidoglycan is good. So having a thick layer is positive. That's what they want, okay? Gram negatives have very little, okay, so I'm kind of abbreviating to save myself some time of writing. Very little peptidoglycan, and that is bad. It is negative, okay, very, very bad, because not having much peptidoglycan means the structure and stability of that cell is at risk, okay? So that's really bad. So they're going to have to compensate for that, and we'll come back to that. We need to make sure that the peptidoglycan for our gram positives stays attached to the rest of the cell, okay? And so it's going to have that teichoic acid, which is going to kind of anchor the peptidoglycan to the plasma membrane, okay? That's not how you spell teichoic. I'm going to put it in the chat, y'all. Trying to write with this mouse is slowing me down way too much. All right, so the teichoic acid, part of its job is to anchor the peptidoglycan to the rest of the cell. So gram positives, that's all there is to it. Teichoic acid anchoring that thick layer of peptidoglycan. Thick is good, positive. Gram negative, that tiny little wimpy layer of peptidoglycan is bad, okay? Having that little of it means they need to compensate for it. And so they're going to compensate for it by having an outer membrane, okay? An outer membrane. This is a second, basically it's a second plasma membrane, okay? It's going to be a phospholipid bilayer. So if you look at the pictures in your PowerPoint, you'll notice that there's two phospholipid bilayers. The normal plasma membrane, and then another one on top of that thin peptidoglycan, because it needs something else that's strong to maintain the shape and integrity of the cell. Now, this outer membrane is super advantageous to the gram negatives because it contains two important things. It is going to have porins, which are little pores, and it has lipopolysaccharide, which is an endotoxin, okay? So the answer to B is gram negative. Gram negative cells are more problematic because they're much harder to treat. Because these little porins that they have, have a very specific shape. So only molecules with that same shape can get through. That's how they make sure only the right stuff gets in. So antibiotics, which the cells don't want, right? It kills them. The antibiotics aren't going to have the same shape as the porins naturally. Yes, Melissa, I will. Okay, so we humans with a good brain, we have had to take the structure of antibiotics and alter that structure in such a way that it mimics the shape of that porin. That way, the antibiotic is tricked into the cell. Okay, and we will come back to this when we get to antibiotics, but porins are why gram negatives are harder to treat, because we have to design drugs that are effective, but also that specific shape, and that's really hard to do. The second one we'll talk a lot about in Unit 3. LPS is what I'll normally call it. It stands for lipopolysaccharide, and it is an endotoxin. And so it's just this built-in toxin that gram negatives have. So when we do kill them with antibiotics, a lot of times the bacteria kind of like blow up, and that releases that toxin into the bloodstream. So sometimes when you first take antibiotics, you feel worse before you feel better, because this little toxin is going in our body. That is why. Yep. Because those porins make it really hard for a lot of antibiotics to work. And so we'll talk about the technique that we use to figure out what can we use to actually help them. All right, y'all. So real quick, sketching a plasma membrane, there's a picture in your notes, but what I really wanted this question to do is remind you of what a phospholipid bilayer looks like. Okay, so there's one phospholipid. It's a bilayer, which means there's two. And they're arranged like this. The word you have to remember, I'm going to put it in the chat because you're going to hear it a lot in this class. A phospholipid is amphipathic. That means part of it loves water, hydrophilic, part of it hates water, hydrophobic. So these circle regions love water. They are hydrophilic. And so that's why they're in contact with water, right? There's going to be water all outside the cell and water all inside. This region right here hates water, hydrophobic. This is why most molecules cannot cross the plasma membrane all willy-nilly, because they're not compatible, right? Water is the universal solvent because most things love water. They're like, water, yes, dissolve me, interact with me, right? They love it. Well, hydrophilic things are not compatible with a hydrophobic environment. And so because this area is not compatible with most things, it lets the plasma membrane be a barrier, prevent molecules from crossing without their permission. So think of the plasma membrane as like the gatekeepers. They're regulating what gets into and out of the cell, because that region of the plasma membrane is incompatible with most molecules. We okay with the plasma membrane? All right, so if you need to go, please feel free to hop off. I'm going to go through this kind of relatively quickly. What the goal of this part was is I've sketched out most of the structures on a prokaryote, okay? And the goal was to have you first, just with your brain, list all the parts of a prokaryote. Because come exam day, you're going to need to know them like the back of your hand, okay? But what I'm going to do instead is I'm going to go ahead and just label them with y'all real quick. So I am going to ask you, hey, what's this? And if you know, put it in the chat. Again, trying not to use your notes if possible. All right, so this black line here, what would that be? That's the border of the cell. What would this be? What is the border of the cell? Hint, it's the gatekeeper that we just talked about. Yeah, it's that phospholipid bilayer. And so we're going to call it the plasma membrane. But you're absolutely right. It's made up of a phospholipid bilayer. Good. All right. Because this is what says, hey, these things are outside the cell. All of this is inside the cell. So you can think of the plasma membrane like the wall. It's the wall separating the outside from the inside. All right. What is this thick yellow region? This is that sugary layer that some bacteria have. It's going to be the capsule. Yes. So remember, the capsule does a lot of things. It helps with attachment because it's sticky. It helps evade the immune system. And if cells are starving and desperate, technically they could eat it, right? But that's kind of weird. Now, plasma membrane, all cells have. All prokaryotes, eukaryotes, everything. If you're a living cell or a living organism, you have a plasma membrane. Capsules are optional. Most pathogens have them, like pathogenic bacteria, but other bacteria that aren't dangerous, they typically don't have a capsule. Because they evade the immune system, you tend to find them on pathogenic bacteria. All right, these little hair-like structures coming off of a bacteria, these are called fimbriae. These are only on bacteria, and they help with attachment. Kind of the more sciencey term I tend to use is adhesion. So these are little hair-like projections that cover the bacteria. They are also optional, but they're only found on bacteria, but they are an accessory structure and they help with attachment. This is going to be the pilus, again, only on bacteria, but it is also an accessory structure, okay? So I'm going to highlight the ones that are absolutely required for life, or at least required for bacteria. In this case, plasma membrane is required for all living things. But so far, capsule, fimbriae, pilus, those are accessory structures. All right, down here, these long whip-like structures, those are going to be my favorite. They are flagella. Now, they are an accessory structure. Some bacteria don't have them, but you do find them on prokaryotes and eukaryotes. Okay, they're optional, but you do find them on both types of organisms. All right, these little dots in here, these are ribosomes. Does anybody know what ribosomes do? And we're almost done. I probably need about 90 more seconds. So ribosomes are protein-making machines. So this is something that all living things must have. They are critical to survival. All living things must be able to make proteins. So you're going to find ribosomes in prokaryotes and eukaryotes. Again, if it's alive, it's got to have them. All right, so this thick pink line, oops, how do I get rid of that? That thick pink line is going to be the nucleoid. Yes, good, Paula. Nucleoid. Now remember, that's all the DNA that is absolutely essential, okay? But another word for this is chromosome, okay? So here's where you have to pay attention to terminology. If you see the word nucleoid, okay, that's all the DNA in a prokaryote. Only prokaryotes have a nucleoid. But if instead I use the word chromosome, chromosomes are in all living things, prokaryotes and eukaryotes. Okay, so if you're alive, you have a chromosome. But in eukaryotes, that chromosome is protected within the nucleus. In A prokaryote, the chromosome, is all just clumped together, hanging out in the region we call the nucleoid. So nucleoid is a prokaryotic term. Nucleus is a eukaryotic term. Chromosome is for both. Are we okay with that? And this is essential for life. Whoops. Why is it not clicking when I'm asking it to click? There we go. Nucleoid is essential. All right, and I think there's only one thing left for me to mark, and that's going to be this orange circle. We talked about this a bit earlier. This is a plasmid. So this is an accessory structure. Remember, it's a bonus feature. It's an extra piece of circular DNA that offers some sort of advantage. It is only found in prokaryotes. Okay, so hopefully this kind of helped you with your week 1 activity. All right, before we wrap up, are there any questions that I can answer for y'all? If you're typing a question, keep going. I just want to remind you, next week, we're going to do a review session over genetics and viruses on Thursday night, Thursday at 7, not on Saturday, because that's exam day and some people want to take it earlier. So we're going to do the review session at 7 o'clock. A lot of people were also available at 8. So if it runs a little late, that's fine. But of course, you can come and go as you need. It will be recorded. So next Thursday, not next Saturday. And if you're going to come, I recommend trying to at least listen to the content for everything that week. Doesn't mean you have to, be a boss at it. There's a lot to keep straight. But with what I review, it's going to make a lot more sense if you've at least heard it before, because it's probably one of the hardest weeks of the class. So, all right, let me see. Can you repeat nuclear? Yes. Okay. All right. So, and if you do need to go again, please feel free. All right. So, Melissa, can you repeat the nucleoid? Yes. Okay. So, what I have sketched here is technically the chromosome. It is the essential required DNA that this organism needs to live. All living things have a chromosome, right? All living things must have DNA because that's the directions that make us, or an ant. So all living things, prokaryotes and eukaryotes, have a chromosome. When you hear nucleoid and nucleus, that kind of means where is that chromosome stored? In a eukaryote, there's an actual compartment, the nucleus, which contains or houses the chromosome. But in prokaryotes, there's no internal structure, right? So all of the DNA, all of that chromosome just kind of hangs out together. And so that region of the cell where the chromosome is referred to as the nucleoid. So the nucleoid is a prokaryotic term. Nucleus is a eukaryotic term, but chromosome is for both prokaryotes and eukaryotes because chromosome equals DNA. Yeah, you're welcome. All right, y'all, that's all I've got. I truly appreciate y'all coming to this review session, and I hope to see you Thursday night at 7. Let me know if you have any questions, y'all. Bye.